Hello everyone, I'm Anas Al-Ubaidi, and the title or the topic for this lecture or session is Foundations is Essential Elements for Developing a Clinical Trial Protocol. So the topic or the title for this session is Essential Elements for Developing a Clinical Trial Protocol. That is part of the chapter that is about foundations of clinical research. And of course, this lecture or session is part of the Advanced Clinical Research Assistant Certification course program by CCRPS. Introduction. Clinical trial protocols serve as the backbone of any medical research study involving human participants or subjects. These documents are not only critical for ensuring that the trial is well organized and efficient, but they also uphold the ethical and regulatory standards that are essential for participant safety and reliable results. A protocol acts as a blueprint outlining every aspect of the trial, from objectives and methodology to data analysis and reporting. It ensures that all stakeholders, researchers, regulators, sponsors, and participants are aligned on the trial's purpose and execution. The importance of a clinical trial protocol extends beyond logistics. It plays a vital role in protecting participants by clearly defining inclusion and exclusion criteria and providing robust risk management plans. Regulatory compliance is also a key consideration, as protocols must meet the requirements of health authorities such as the FDA in USA, so US Food and Drug Administration, EMA in European Union, European Medicines Agency, and other global bodies similar to EMA and FDA, depending on the geographical area, like MHRA, MHRA in UK, United Kingdom, or TGA, Therapeutic Good Administration in Australia, and so forth. So, and other global bodies to ensure the trial is legally and ethically conducted. For clinical research assistants, CRAs, understanding the intricacies of developing a protocol is paramount. From concept development to execution, every detail must be meticulously planned and documented. Key points. We'll start with the objectives and hypotheses. The foundation of any clinical trial lies in its clearly defined objectives and hypotheses. These elements establish the purpose of the study and guide every subsequent decision from design to data analysis. Purpose. Objectives articulate what the aims to achieve, such as evaluating the efficacy or safety of a treatment. Hypotheses provide testable assumptions based on these objectives. Example, an objective might focus on reducing blood pressure using a novel drug. The hypothesis could predict uh, uh, that this drug is more effective than current treatment. So, this is a simple, perfect example. An objective might focus on reducing blood pressure using a novel drug. The hypothesis could predict that this drug is more effective than current treatments. Significance here: well defined objectives and hypotheses ensure that the trial remains focused and scientifically valuable, avoiding unnecessary deviations or inefficiencies. And here is a diagram on foundation of clinical trials. Another key point here: inclusion and exclusion criteria. Deciding who can participate in a clinical trial is a crucial step that directly influences the quality and applicability of the findings. Definition: inclusion criteria specify the characteristics participants must have, while exclusion criteria identify those aspects that disqualify individuals due to potential risks or confounding effects. Example, a cardiovascular drug trial may exclude patients with severe pre-existing conditions such as advanced heart disease to minimize risk while ensuring the results directly pertain to the target population. Impact: these criteria ensure the participant population is homogeneous enough to yield reliable results but still representative of the broader patient population. And here is a diagram on the role of criteria in clinical trials. Another key point: detailed description of trial phases. Clinical trials are conducted in multiple phases. Phase 1, 2, 3, 4, each with distinct purposes and methodologies. A protocol must outline these phases in detail to provide a clear trial pathway. Phase 1 focuses on safety and dosage levels in a small participant group. Phase 2 expands to assess safety and efficacy in a larger group. Phase 3 compares the new treatments to existing ones or placebos in diverse populations. Phase 4 monitors post-market safety and effectiveness in the general population. Example, a cancer drug protocol might detail plans for small-scale phase 1 trials to identify safe dosing prior to moving into larger phase 3 studies to confirm efficacy across multiple sites. Enhancement here, descriptions of each phase help ensure trials follow logical progression, providing clearly defined endpoints and key performance metrics for each stage. And here is a diagram on clinical trial phase progression. Another key point here, interventions and assessments. All interventions, for example, drugs, devices, or behavioral changes, and assessments, for example, laboratory tests, imaging studies, must be explicitly detailed in the protocol. Precision here, clearly specifying dosages, administration methods, and timing ensures consistency and reliability across study sites and participants. Example, a diabetes trial might include detailed instructions for drug administration, such as 10 milligram oral dose twice a day for 12 weeks, and specify measurement intervals for glucose levels. Significance, proper documentation minimizes deviations and ensures the trial produces reproducible results. And here is a small, simple diagram on precision and significance. Ethical considerations are as a key point. The protocol must address ethical requirements to protect participants and ensure the study complies with international ethical standards such as the Declaration of Helsinki and ICH GCP guidelines. Risk minimization includes strategies to monitor and mitigate risks such as regular safety committee reviews to evaluate adverse events. Example, implementing safety mechanisms like interim analysis enables researchers to halt trials if risks begin to outweigh benefits. Informed consent, the protocol must outline procedures for informed consent, ensuring participants fully understand the study's purpose, risks, and benefits. Example, providing clearly written consent forms supplemented by verbal discussions ensures comprehension, especially for populations with limited literacy. Impact here, ethical adherence not only protects participants but also enhances the trial's credibility and acceptance on a global scale. And here is a diagram on ethical framework for clinical trials. Another key point here, global impact and standardization. To ensure the validity and global applicability of study findings, protocols must align with international standards and regulations. Standardized practices protocols should adhere to internationally recognized guidelines such as ICH GCP, International Council for Harmonization, Good Clinical Practice to promote uniformity and quality. Regulatory compliance, meeting the requirements of agencies like the FDA in USA or EMA in European Union, ensures that the trial is authorized and credible. Example, a multinational trial for a rare genetic disorder uses identical assessment protocols and data collection methods across all sites, enabling valid comparisons of results. Significance here, standardization fosters global collaboration, increases acceptance of findings, and streamlines regulatory approval across regions. And here is a diagram on ensuring validity through international standards and collaboration. Another key point here, examples of clinical application. Real-world examples help illustrate how well-constructed protocols contribute to the success of clinical trials. Example 1, a diabetes drug trial uses strict inclusion criteria. For example, fasting glucose levels more than 140 mg per dl to ensure an appropriate participant pool. Example 2, a cancer trial prioritizes standardized evaluation methods, for example, tumor size via MRI across multiple countries to ensure data comparability. Example 3, trials addressing COVID-19 vaccines adhere to global ethical and regulatory standards, such as publishing results transparently to facilitate rapid, widespread adoption. Closing thoughts. Developing a comprehensive clinical trial protocol is an intricate process that demands attention to both scientific rigor and ethical principles. It requires collaboration between researchers, sponsors, and regulatory bodies to ensure trials are safe, valid, and compliant with global standards. For clinical research assistants, mastering the art of protocol development sets the foundation for successful trial execution, ensuring that both the interests of participants and the advancement of science are upheld in equal measure. Here we have the diagram on enhancing clinical trials through rigorous protocol development. Summary tables. Here we have a table on objectives and hypotheses, table on inclusion and exclusion criteria, table on trial phases, table on ethical considerations, table on global impact and standardization. Then application table, as we see here, scenario for each scenario, key principle applied, key actions, timeline, what to watch out for. MCQs, multiple choice questions in order to revise what we have covered through this lecture. Like question 1, what is the main purpose of clearly defining objectives and hypotheses in a clinical trial protocol? Is B the most appropriate answer as to establish the trial's purpose and guide its methodology, as objectives and hypotheses lay the foundation for a clinical trial, clarifying its purpose and setting expectations for outcomes. Question 2, why are inclusion and exclusion criteria critical in clinical trials? As C is the most appropriate answer to select an appropriate and safe participant population, as these criteria help identify suitable participants who can contribute meaningful data while minimizing risks to their safety. Question 3. Which of the following correctly describes the focus of phase 1 clinical trial? We know phase 1 clinical trial, in phase 1 it's about determining the dose and the safety. So C would be the most appropriate answer, testing safety and determining appropriate dosage in a small group, as phase 1 trials aim to evaluate the safety profile of a drug or intervention and determine the optimal dosage. As A, comparing interventions to standard treatments will be phase 3, monitoring long-term safety and effectiveness will be phase 4, assessing efficacy in a larger patient population would be phase 3. So C would be phase 3 as well. So A and D phase 3, B would be phase 4, and of course C is phase 1. Could be D as well, assessing efficacy in a larger patient population, phase 2 as well. So A, comparison is phase 3. B would be phase 4. C is the answer here for this question, phase 1, assessing efficacy in a larger patient population. Larger population in comparison to the population in phase 1. So D would be phase 2. Question 4. What is the significance of including ethical considerations in clinical trial protocol? Most appropriate answer B, to comply with legal and moral standards for participant safety, as ethical considerations like risk minimization, informed consent procedures, and safety monitoring are crucial to protect participants and uphold international guidelines like the Declaration of Helsinki. Question 5. How do standardized practices benefit global clinical trials? B would be the answer, by minimizing variations in trial designs across locations. As standardized practices such as adhering to ICH-GCP guidelines ensure consistency in data collection, trial execution, and reporting across international sites. This promotes comparability of results and accelerates global acceptance of findings. And here we come to the end of this lecture. Thank you very much, and goodbye.